Hi, how are y'all doing? I hope all is well. My name is Brandon Sorrell, and I'm an undergraduate student at Mississippi State University. I am a senior in the College of Wildlife, Fisheries, and Aquaculture with a concentration in aquaculture, and I am originally from Marietta, Georgia. And what brought me to Mississippi State um, was my love of fish. Actually, from a young age, I generated that love, but it wasn't until high school that I realized I wanted to artificially breed fish and farm and cultivate them for conservation, consumption, and private stocking purposes. And this is my project on the environmentally adaptive gills of the alligator gar. All right, so a brief introduction. The alligator gar are bimodal breathers, which basically means they receive oxygen from the water using their gills and from the atmospheric air using an air breathing organ called the swim or the gas bladder. They are facultative air breathers, which means they rely on air breathing when the dissolved oxygen in the water is depleted and as temperature increases. As y'all know, that has an inverse relationship. As temperature rises, dissolved oxygen decreases. And they have a broad environmental tolerance. So this bimodal breathing characteristic allows them to survive in low oxygenated fresh water, like stagnated water in the backwoods, and also salt water. And what's very interesting is they are found in the Trinity River. They are very prevalent in the Trinity River, and as y'all know, that's a river in Texas, and it gets hot in the summertime. So you'll see them come up and gulp air, and that is them utilizing that air breathing organ because the dissolved oxygen is depleted in the Trinity River. There's very little known about the specific physiology and morphology of the alligator gar's gills. Chloride cells, which are cells that are found in the gills, and I will go into more details about them later in the presentation, are mitochondria dominant cells. These cells play a vital role in maintaining internal osmoregulation, ionic regulation, and acid-base regulation. All right, so my research hypothesis: certain cells in the gills assist in the urilinity of the alligator gar, and urilinity or urihaline is just the tolerance for a wide range of salinity. So the objective: catch and separate several alligator gar from the original freshwater tank and acclimate them to saline water, 20 parts per thousand, and extract the gills of a freshwater and a saline acclimated gar to prepare them for scanning electron microscopy and transmission electron microscopy, and then compare and contrast the morphology of the cells and other anatomical structures between the freshwater and saline acclimated gills. Okay, so the methods. Two different 3,500-liter tanks were used in this experiment. One freshwater was zero point 0.1 parts per thousand supplied by well water, and the other was a saline tank, 20 parts per thousand salinity, and it was in a recirculating aquaculture system or a RAS. And both tanks had fish from the same source. 6 fish were transferred from the freshwater tank to the saline tank. Fish in the saline tank were gradually acclimated to increasing salinity at a rate of 0.64 parts per thousand a day by adding artificial sea salt instant ocean. And this took roughly 2 weeks for us to get it to get it up to 20 parts per thousand salinity. Prior to sampling, alligator gar were placed in the anesthesia or anesthetics, which was called the MS222 solution, which is 250 milligrams per liter of tricane methane sulfonate, which was buffered with 400 milligrams per liter of sodium bicarbonate. Okay, and due to the thickness of the ganoid scales, a bolt cutter was required to remove the operculum. Then surgical scissors, a scalpel, and forceps were used to remove the pseudobranch and 4 gill arches individually. Gill arches were briefly immersed or submerged and stirred in a 9 gram per liter of sodium chloride solution to remove adhering blood. For each gill arch, a scalpel and forceps were used to separate the filaments from the gill arch and then to carefully separate filaments and lamellae samples. SEM samples were placed in a 15 milliliter sample vial, and TEM samples were placed in a 2 milliliter sample vial at a 50 to 1 ratio of fixative to tissue. and the fixative was 2.5% paraformaldehyde, 2% glutaraldehyde, and it was buffered with sodium calcodite. And all the fixative does is just it stops the activity of the soluble proteins in the cells, and it preserves basically what the cell looked like before dissection. Samples were stored in the aldehyde-based fixative for 2 hours at room temperature and were transported to the imaging center's lab for the chemical wash process. SEM samples were washed twice in 0.1 mole of sodium calcodite buffer for 3 minutes. The samples were then post-fixed with 1% osmium tetraxide in 0.1 mole of sodium calcodite buffer for an hour, which turned the samples black. Uh, and as y'all know, osmium tetraxide is toxic in the gaseous and the liquid phase. So after carefully transferring the osmium to the waste tube, samples were washed twice in the 0.1 mole of sodium calcodide buffer for 5 minutes. The samples were then dehydrated using a gradient of ethanol. Um, they were washed twice for 5 minutes in the 30%, 50%, 70%, 80%, 85%, and the 95% graded ethanol. And then the samples were washed twice for 10 minutes in the 100% graded ethanol. After dehydration, the SEM samples were placed in 4 little green wafers, one wafer per Gill arch, that were in a petri dish filled with 100% graded ethanol. The green wafers were placed in the critical point dryer at negative 24 degrees Celsius, which is roughly negative 11 degrees Fahrenheit, for 45 minutes. The critical point dryer dries delicate samples using high pressure and low temperature. Once the samples were done drying, they were kept in the little green wafers overnight. The TEM samples were washed in a mixture of ethanol and propylene oxide first, then 100% propylene oxide, and then were soaked in an epoxy resin and propylene oxide mixture. Propylene oxide has a low surface tension and a very low viscosity due to its low friction molecular makeup. The next day, the SEM samples were coated in platinum dust for conductivity using the EMS 150T ES sputter coater. So the results, this is a image of the sample. It's a broad image of the sample. As you can see on the top right side, you can see the 2 hema branches, and you can see the septum that holds them together. And then down the right side, you can see the lamellae projecting off the left side. And then on the left side, you can see the filament running down the middle, and you can see the lamellae projecting off of both sides. And in this picture, which is a more magnified or a close-up picture on the filament and the lamellae, you can see the filament running down the middle. You can see the lamellae projecting off both sides. And then you have the freshwater chloride cells. You can see the numerous microvilli extending outward and upward. And you can see they kind of look like sea anemones a little bit. And then in this picture, you can see the recessed chloride cells. They are called apical pits or chloride channels. And this is in the saline acclimated gills, and you can see the button cells as well on the right side. And in conclusion, these findings support my hypothesis on the morphological differences of the chloride cells. The general morphology of the alligator gar's gills is identical to many teleos fish species, 4 gill arches with 2 hema branches per gill arch. In freshwater gar, there were chloride cells with numerous microvilli extending outwards. In saltwater gar, chloride cells were recessed with apical pits and button cells present over the filament surface. And these findings prove or elucidate the plasticity of cellular gill responses to external salinity change in alligator gar. And there are 3 factors that play into the morphological differences that we saw between the freshwater gar and the saltwater gar. And the 3 factors are acid-base regulation, osmoregulation, and ionic regulation. So since the ocean is more acidic, or saltwater is more acidic than freshwater, the surface area of exposed chloride cells decrease in saltwater due to an acidic environment, which is acidosis. And as the chloride cells decrease in size, or decrease in surface area, adjacent pavement cells expand, causing the chloride channels that that were seen. And during alkalosis, the surface area of chloride cells increase and actually have known to proliferate in certain species. Then you have osmoregulation. Chloride cells adapt to increase salinity by responding to a rapid signal that stimulates chloride secretion, which also forms the chloride channels that we saw. And ionic regulation. During the saltwater acclimation, there's a reduction in the number of functional chloride to bicarbonate exchangers. And here are pictures you see on the left. That is a critical point dryer that we use. And in the middle, you have the alligator gar right before being dissected. And then on the right, you have me doing the chemical wash process. And special thanks to Dr. Peter Allen, Dr. Orion Rivers in the Mississippi State URSP program, Mississippi State Imaging Center, the South Farm staff, and the graduate students that helped me along the way.